सभी को प्रणाम। मैंने अपनी लास्ट वीडियो में आपको ये बताया कि आज डॉक्टर्स और साइंटिस्ट किसी भी बीमारी को अर्ली स्टेज पे डायग्नोज करने में और डॉक्टर्स उसको ट्रीट करने में असक्षम क्यों हैं, और इस धरती पर एक ऐसा दिव्य स्थान मौजूद है जहाँ ये काम बिना किसी परीक्षण के, बिना किसी व्यक्ति को छुए सिर्फ 5 मिनट में किया जा सकता है। तो पहले हम साइंटिफिक अप्रोचेस को, जो आज रिसर्च पेपर्स में पब्लिश्ड हैं, किसी एक डिज़ीज़ के माध्यम से समझने का प्रयास करते हैं। फिर धीरे धीरे हम इसी चीज़ को स्पिरिचुअल एस्पेक्ट से भी देखेंगे, और फिर हम ये कैसे संभव हो पा रहा है उस जगह पर, इसको समझने का प्रयास करेंगे। तो सबसे पहले मैं आज इस बात को एक्सप्लेन करने के लिए आई कैन जस्ट टेक एन एग्जांपल ऑफ अ वेरी कॉमन डिजीज़ विच इज़ कैंसर। आज हम यदि ये कहें कि कैंसर जैसी बीमारी जिसमें देयर इज़ एन अनकंट्रोल्ड सेल डिविजन, दैट इज़ अ मेजर चैलेंज वर्ल्ड वाइड, एंड द पीपल आर not able to find any relief after getting all the possible treatments. आपको मैं थोड़ा सा ये भी बता दूं कि द साइंटिस्ट बिलीव कि कैंसर इज़ अ डिजीज़ इन विच देर इज़ हाई ऑन्कोप्रोटीन प्रोडक्शन। दे बिलीव कि विद इन द ह्यूमन बॉडी our genetic material, which is the DNA, and all the proto-oncogenes within the DNA sequence, they contain some G-rich segments that allow the DNA to fold into higher order DNA structure. In the promoter region, these sequences are present, like for example, and the higher order structure which is formed by the G-rich sequences is the G-quadruplex. So this G-quadruplex is a 4-stranded DNA structure that allows or that controls the rate of gene expression. Means कि we have and cancer causing genes within us, which we call it as proto-oncogenes. So after the genome analysis of the human, all the proto-oncogenes, they have been identified, and researchers have already identified and published कि like the human proto-oncogenes, their promoter regions, they are found to be G-rich. And these G-rich sequences, they allow the DNA to fold into higher order DNA structures, and that structure, when it binds with the protein, it completely regulates the rate of transcription and the rate of translation, and as a result, the quantity of the oncoprotein is very high. Whenever we take any drug, we have to understand why these kind of diseases, they comes under the category of the incurable diseases, right? And these kind of diseases, they cannot be treated by giving the drugs, because whenever you take any chemical drug, that drug ultimately bind to the some targets within the cell, and generally these targets are protein. But in the cancer patients, the cell is in immortal state, there is a high oncoprotein production, and as a result of which, when you take the drug, the drug, it has a limitation, right? When you will consume the drug in excess, then it will be difficult for the body to remove the drug by the drug metabolizing pathway. You might be familiar that we have a drug metabolizing pathway which is called cytochrome P450. So when you will take the drug in excess, that drug cannot be metabolized properly, and once it will accumulate inside the cell, then it will become toxic. So toxicity will be the problem, and hence they are trying to treat the disease or the tumor by the chemotherapy. Chemotherapy is nothing, it is a kind of the radiations using which you are burning the cell, but you yourself can think how the radiation can identify the specific cancer cell and a non-specific cell. So when you are exposing the body part with the radiation, the some non-specific cells, they are also killed, and hence the people are suffering from lots of side effects. The major challenge for the researchers is to identify the cancer at an early stage. When the cancer comes in a 3rd stage or a 4th stage, then they are able to identify the diseases, and then that time it is very late, because it might be possible that once the disease can be diagnosed at an early stage, you can just relieve the patient by giving the some possible or the alternative drug, and hence disease diagnosis is a major challenge. But what are the possible approaches are available? I will just discuss in brief. They used to do the biomarker identification or the identification of the small molecules in the body fluids. because whenever the person suffers from any disease, there isn't some abnormal secretions or the some abnormal or high concentration of the molecule are present in the body fluids, right? And secondly, some chemical biomarkers in a DNA. I can just give an example because in case of the DNA sequence, there are 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. But when the person suffers from the cancer, that time there is an abnormal production of abnormal appearance of A-toxoguanine. A-toxoguanine, 2-aminopurine, these are the chemically modified form of the nitrogenous bases which are present in the DNA sequence, and based on the population studies, they are identifying these biomarkers, and based on the identification, they are able to predict the disease. Okay, like if this biomarker is present in a DNA sequence, so there are chances that that person will have this kind of disease. So these are the possible approaches. But to be very frank, although there are lots of papers which are published on a biosensors in the research community, but I do not think that there is any true biomarker which can identify the real concentration of any molecule inside the cell, right? Or the real change in a DNA sequence inside the cell. Not only this, the scientists are failed to understand that like we all the humans, we look similar in our appearance, in our genotype and the phenotype. But to be very frank, if you understand the human body deeply from the energy viewpoint, from the spiritual viewpoint, then only you will be able to understand that each and every individual is very, very unique. And hence, you cannot give a single medicine to all the person. If it was true, then suppose a person is suffering from a fever, there must be a single medicine for a fever. But no, if there are 10 people, those who are suffering from the fever, and you are just giving a single medicine, it may be able to work on a 1 or the 2% and rest of the 8 people, they will still suffer, right? So we have to understand like these approaches, why they are not able to give any solution for the complex illnesses. The third approach on which the scientists believe about the cancer is telomere dysfunctioning. Telomeres are basically the short single-stranded DNA which are guanine rich. They are single strand, and these are ultimately the caps of the chromosome. I just wanted to tell you that like our DNA, the length of the DNA is 3 billion base pairs, which is present in the form of 23 pairs of chromosome within us, and it is present in the form of nucleoprotein complex. But at the end of the chromosome, there is a long tail. Like within the human, we have an hexanucleotide which is called GGGTTA. So this hexanucleotide repeat is found to be 250 or 300 times longer as a single strand, right? And the scientists believe that like whenever the cell undergoes the division or the replication, that time there is a different mechanism in a germline cell, which is an immature form of the cell. But when the same cell turns into the somatic cell, which is a mature cell, then there is a different mechanism. So here we have to understand the role of the telomerase enzyme. You have to understand like cell is a basic structural and the functional unit. This we know very well. It is very well documented in the books and the research papers. But when the cell grows and then it combines, it forms a tissues. When the tissue grows and combines, it forms an organ. When the organ having a similar functions, they assembles, then it forms an organ system, and the different organ systems they makes a one living individual. But the ultimately cell is a basic structural and functional unit. So each and every cell has a certain lifespan. हमारे अंदर क्या है कि cell death, which we call it as apoptosis, it is a continuous phenomena. So the cell dies maybe after a few days, after a few months, after a few years. The cells are not immortal. Cell, which is a basic structural and functional unit of any human body, is not a permanent cell. It will die by the process of cell apoptosis. There is a defined mechanism, and as a result of which, when the cell will die, then automatically in our bone marrow there are hematopoietic cells, and as a result of which the cell also regenerates. So there is a regeneration capacity also, right? So whenever the new cell forms, that time the body operates differently. But when the same cell turns into the somatic or the mature cell, then the body operates differently. So in a immature germline cell, they have identified that there is one enzyme which is called telomerase. telomerase is basically a ribonucleoprotein complex that contains RNA as well as a protein part. The RNA part is TER and a protein part is TERT, right? So whenever the DNA, because DNA is a master molecule, it ultimately regulates the genetic material. So when the DNA replicates, that time there are two strands in the DNA, so there is no problem in the replication of the one strand because the DNA polymerase, which is a main enzyme, it is found to be directional specific. So as a result of which it will keep on adding the nucleotides in a directional specific manner in a 5' to 3' direction. But the problem of the replication comes in the another template strand where the polarity is different. So when the template is itself is 5' to 3', then DNA polymerase cannot add the nucleotides in a 3' to 5' direction because it is directionally specific. And as a result of which the another strand folds into the loops, and that gives the same direction to the DNA polymerase enzyme, which can allow the polymerase to add the few nucleotides, and as a result of which the second strand of the DNA forms in small fragments, which we call it as Okazaki fragments, and later on these fragments they are ligated together by the ligase enzyme. But the problem comes in an end. Ends mean there is no sufficient length for the DNA which can allow the DNA to fold, and as a result of which there is this is a end replication problem, is a major problem. But this problem is overcome in germline immature cell by another specific enzyme which we call it as telomerase. So as the telomerase is a ribonucleoprotein, so presence of this enzyme in axis allows to add the short stretch of the RNA at the ends. And because the protein is a reverse transcriptase, so it uses the RNA as a template and allow the RNA strand to make a DNA copy. So as a result of which there is no shortening of the telomere in the immature cell. So this process is completely common in all of us. But after the some successive replication, when the same germline cell turns into the somatic cell, that time automatically the expression of the telomerase enzyme, which was responsible for to overcome the end replication problem, that concentration goes down. So when the telomerase enzyme is found to be not present in a somatic cell, so then automatically after every successive replication, because cell is replicating continuously, the somatic cell is also replicating the DNA molecule, but now it is replicating the DNA in the absence of telomerase enzyme. So as a result of which whenever the DNA replicates, it shortens the DNA sequence around 50 base pair, 50 bases, and then 30 to 40 or 30 to 50 bases, and then there is a continuous telomere shortening. This telomere shortening is a major cause, like with an age we are moving towards the old age, right? So why we are just moving towards an aging? Because telomere shortening is a major cause which is identified by the doctors or by the researchers, right? The telomere shortening is one of the cause. Because the telomerase enzyme which can overcome this end replication problem, it is not present in the mature cell, and when this mature cell grows further, then it will keep on replicating the DNA. And then automatically we have a mechanism कि when the length shortens, it is a successive process. So when the length is short, around 30 to 40 or 50 base pair, then automatically it gives the signal to the cell to stop the DNA replication. This process is called M1 phase, cell motility state, senescence. And automatically there is a mechanism, another enzyme which is called P53, which is a tumor suppressor enzyme, it gets activated, and it does not allow the cell to replicate, DNA to replicate further. But suppose if the the tumor suppressor enzyme P53 is found to be mutated or altered somehow, just because of any reason, then the cell keeps on replicating the DNA, and as a result of which there is further shortening of the telomere that ultimately lead to the M2 state, crisis state, and it lead to the massive genomic instability. So there are lots of genetic diseases nowadays which we cannot, not only we can identify, in fact we have no solution or the no cure of this kind of disease. We cannot think about it. In fact, there is no scientific literature which is available to give any problem to the modern society. So the problem is कि in the cancer patient, the point is कि telomerase enzyme is found to be active in a mature cell, and as a result of which the DNA does not undergo the apoptosis. So cell apoptosis is not taking place. DNA replication is not going to end, and as a result of which the DNA will keep on transcribing and translating, and ultimately there is a high oncoprotein production. So that's why the researchers are working on the telomerase enzyme, although the point to be noted, they do not know the reason why the telomerase enzyme, which has to switch off now, why it is switch on in the somatic cell of the cancer patients. So that's why the cell is continuously growing. There is no cell death. In fact, the cell is attaining the immortal state. Right? And then based on the some biological targets, they are using either the telomerase enzyme itself or the DNA sequence on which the telomerase is going to bind as a therapeutic target, because the telomere itself is a G-rich overhang, which is GGG-TTA. It is an hexanucleotide which is found to be repeated 250 or 300 times in all the individuals, and enzyme telomerase is structure specific. So it can bind to the single stranded G-rich overhang, but it cannot bind to the higher order structure which is formed by this single strand nucleotide, which is called G-quadruplex. So that's why they are working on the small molecules which can bind to the G-rich overhang and which can bind to the G-quadruplex. They can stabilize the G-quadruplex, and as a result of which, because telomerase enzyme can bind to the single stranded G-rich overhang, but it cannot bind to the G-quadruplex, which is a 4-stranded higher order DNA structure. So that's why they are identifying the small molecules, they are designing the small molecules or chemical drugs, or they are isolating the natural molecule from the plant sources, from the microbes, and then they are synthesizing these molecules in the laboratory. And then after the successive biophysical studies and the same testing of the drug in the cell lines in different cell cultures, and then in the animal models, then the same drug goes to the clinical trials, and then it comes to the market. But when the point we have to understand is that when each and every individual is unique and different, then how this kind of medicine can provide any solution to treat any kind of complex illnesses, right? So I have discussed in this video about all the possible approaches which are available or which are already published in the research papers. So there are lots of research papers on which the telomere dysfunctioning or the upregulation or the dysregulation of the promoter regions, and what kind of drugs are available, about the chemotherapy. All this literature is available. You can just put the specific keywords in a Google and you will be able to download many research papers, right? So it is very well documented whatever I have discussed here. Now we have to understand, in the next video I will tell you first what is the reality of the human body. We all look like a human, so the entire human population, how it is different from each other with respect to the point of energy, right? Because you might be familiar whatever the energy being we are radiating the energy in the form of energy aura, and each one of us is radiating the different kind of energies, right? So how this energy is emitting, how the aura is developing, this we will going to understand in the next video. Penda.